All right, so really only two lectures, this one and the next one, before the review lecture. So schedule here, looking forward. So the timeline, this is a little bit off because I timed this when we don't have Veterans Day. So I normally give this lecture on Tuesday. So the spacing is a little bit off, but it's basically about the same here. So we've got, ICAL is available. Homework J2 has been posted. I know some folks have been working on that. The lab. So technically, there's the Lab 10 was due, this past weekend, but I make it available because it has this extra bonus. Normally, I make it available until this upcoming Sunday, but because of Veterans Day, kind of it skipped a lab period and so on. Now it's available through Wednesday, so it is reflected on Canvas. If you happen to be using the old textbook or a copy of an old textbook, or even if you're watching that on a lecture video I had, which was recorded before I decided to start using the new version of Arena for this, then if you're looking for schedules, signal, and hold, it will tell you to go into a different panel. It'll tell you to go to either the advanced process or the advanced transfer panels for those. I've added notes to the PowerPoint presentation, as well as both the Lab 10 report page on Canvas and the Lab 10 lecture page on Canvas, that tell you that to make sure to use the signal, hold, and schedule from the new panels in Arena. So don't go and load those old advanced transfer and advanced process panels. Grab those from the decisions panel and the data elements panel. I forget what it's called, like whatever the red panel is that all the data modules in at the very top. That's where the schedules are. So if you try to load the model, which I've, as of this semester, I've now used the models from Arena 16.2 for that model 7.1. If that model 7.1, it's using schedules, but it's using the new schedule block. So if you look for schedules when the old place, it'll say there's no schedules located. And so, and no routing data located. And so in that case, you need to look for that in the new location there. So all of that is discussed again on the main pages. But if you watch the video, I may still in that video refer to advanced process, advanced transfer, but look on the actual Canvas page or in the actual PowerPoint presentation, and I'll give instructions on where to find those. And they're they're relatively easy to find. They're in the standard panels. You don't need to load anything new. So that's just a hint there for that Lab 10. Otherwise, no more labs. So it's just working with your final project. So the final project here, the big things here is the timing. Presentations are due the Wednesday, the last week of class. Final report due Wednesday, last week of class. They both have got a format that's specified online. The presentation, no longer than 10 minutes. It's fine if it's shorter. You don't have to use all of your team members in the presentation, so you don't have to have like equal time or something like that. And so I recognize that some team members are going to be helping more on the back end. Some team members are going to be helping more on the front end. So that's fine. You could use PowerPoint, Keynote. You can use Arena animations. You can use recordings of Arena animations. All of those things are fine. You can swap over. You can Alt Tab from PowerPoint into Arena during your presentation. That's fine. Presentation grades are determined in part by peer review. So after you upload this, then after Wednesday night, then I'll go ahead and tell Canvas to auto assign randomly another team to you for you to evaluate their presentation and their final report. You'll go and fill out a rubric, fill out some comments, submit that before the Saturday before finals week, and then I'll aggregate those scores and then watch everything myself so that we can get a final score that's the combination of those two. So that's the final project there. There's still about, I think, 2 input modeling reports that I haven't hit submit on the grades for. It takes me longer than you might imagine to get all of those graded and to try to give useful commentary there. But there's no input modeling reports that I've seen that made me think that people should go in a completely different direction. If you do have specific questions about your final project, regardless of whether you've gotten your input modeling report back, just send me a note and we can address that. Whether you need to make things a little more complex. In most cases, you just simplify it. Like most groups said, we're going to study all these different things. And in my feedback, I said, just pick one. So, you don't have much time left. It's just an A versus B, a kiosk versus a cashier. You've got a particular way to make a sandwich versus a particular other way to make a sandwich. Those those two things, usually as is and proposed. That's all you need. You don't have to necessarily optimize over a bunch of degrees of freedom. There's not just a time for that. Otherwise, final exam review. So if you do that lockdown browser compliance test for the final exam, it'll unlock the final exam module, and you'll be able to find a bunch of useful resources in there. And so, take a look at that. The final exam is exactly the same structure and length as the midterm. Because stage one is designed to fit within a class period, it's the same length as a midterm. It is cumulative, but we're going to bias things to post-midterm. But if you think about that, a lot of the stuff post-midterm have been kind of elaborations of pre-midterm. Like we talked about the chi-squared analysis pre-midterm, but then we made it more general post-midterm and those sorts of things. So, the final exam, you are going to need to know how to do a hand simulation. Expect to see a couple of arena diagrams and maybe be asked questions about those diagrams on the final exam. There's no practical, like I'm not going to ask you to build anything and upload anything in Arena. Everything's auto-graded, so it is very midterm-like. It is a two-stage. So basically in the last week of regular scheduled classes, Tuesday will be the review lecture, Thursday will be stage one, which if you don't have to come here, but if you want to come here, I'll be here staffing the room. So if you want to take it here in a quiet space, that's fine. But I'm going to make this available. I think I said last time I'll make it available stage one. Maybe I got on the next slide. No, I think I've said I'll make stage one available like Wednesday, Thursday, Friday, and then stage two available Monday through Friday of finals week. So stage two, I will be here during the regularly scheduled time. It's on Canvas. I think it's like 9:30 or something on Tuesday of finals week. So the room will be staffed if you want to come to stage two, the collaborative portion, but you don't have to, just like the midterm. And that I'm going to make available all finals week. Monday through Friday, just like the midterm, you won't have your scores in between the two stages. You won't have answers in between the two stages. It's exactly the same as the midterm. I do give you one extra sheet, I think, in your formula sheets. So that's one difference. Any questions? Yeah. The plan is to open it Wednesday, Thursday, and Friday of the last week of classes. So it's technically, in both cases, the due date on Canvas, just to get it on your schedule and to give you my target for when I think it'd be great if you had it done by. The due date will be Thursday, but it'll be open through Friday. So you get those 3 days that you can do those stage one. You get the weekend off. And then likewise, the due date for stage two will be Tuesday of finals week, but it'll be open with no penalty Monday through Friday of finals week. And it's all Canvas based. Lockdown browser for stage one, no lockdown browser for stage two, just like the midterm. So you want to take it remotely, that's fine. If you have travel plans and you'd like to take it elsewhere, that's fine. Okay. All right. So, we've been, culminating in interval estimation and using these confidence intervals to make sense of our data. So that's the type of analysis I'm going to hope to see in your final project report, where you're going to give me estimates in terms of confidence intervals, and then you're going to be making claims about those confidence intervals that, these two systems are different, and these two systems, I can't tell the difference. They don't have much of an effect. So we're trying to get you out of this point estimator habit. So break the point estimator habit, reference to Point Break, at least when it was made, when I was of movie watching age. It's been remade, so this is probably a unrecognizable reference now. But this is what we talked about last time, where we said that these confidence intervals, you frequently use them to look at relationships, and so that's where we've talked about two sample confidence intervals now. And so we talked about with linear regression, that's an example where these slopes that you fit through lines, The slopes themselves are uncertain quantities. So you don't actually know the exact slope of this relationship. So people use confidence intervals around the slopes of these relationships, around these regression coefficients. And those confidence intervals, they get plotted here. And if they exclude certain important points, like it's often you ask, does it exclude zero? Because if it excludes zero, that means there is a relationship. Because if it includes zero, then it's possible there's no relationship between those two variables. So it's a flat line, it's a horizontal line, in which is no relationship. But if it excludes zero, like this one, then there is a relationship. So then you can start asking questions about what type of relationship is it? Is it in this case, this is, so the fact that this is log-log means that this is an exponential relationship if you unlog them. And so when you log them together, you end up getting a line. And so this slope corresponds to the exponent of that exponential relationship. And if that exponent happened to be one of that relationship, you would get sort of a flat line, the way this thing is written. And if it's less than that, it bends down. And if it's greater than that, it bends up. And so what this is saying is that it bends down because all of these numbers are less than one, but exactly how it bends down, it could be any of these numbers, but there is a mathematical model that suggests it should be 0.75. And this says, well, that is reasonable. So this does not reject that model. So you can tell a lot from a confidence interval by what's in the interval and what's out of the interval. If they just had given us a point, if they just gave us that 0.7845, then that doesn't tell us a lot. You can say it's close to 0.75, but is it close enough? It's also arguably close to 1.0. So the confidence interval tells us not only is it close, but there's pretty much no way the actual slope could be 1.0. There's no way the actual slope could be zero, but it definitely is reasonable for the actual slope to be 0.75. So that's how we read these confidence intervals. And we apply that to figuring out the absolute performance of systems. So, we want to say, what is the wait time for callers? And so, is it 5 minutes? Is it 6 minutes? And so you can kind of confidence interval for wait times. And so that's what, this looks like here. So you're going to have a one sample confidence interval. So these here for your performance variable, like average wait time, these are all of the wait times that we cannot exclude. So we're saying based on their samples here, if your performance target is inside this interval, then that means we might have met your performance target. If you have an absolute maximum wait time, and that's what, so like if you've got a wait time target that's outside of this interval, then that tells us that you're definitely under your target or you're definitely over your target. So this gives us an idea of the error of the estimate so that we can make claims here. So if something's inside the interval, there's ambiguity. If something's outside the interval, it's pretty clear that the estimate rejects that possibility. So the way we calculate these confidence intervals is we're just basically visualizing a t-test, but it's not very stakeholder friendly to then have t-test results over and over again. So we give them confidence intervals instead, but it's exactly the same thing. We just don't call it a t-test, we call it a confidence interval. And so we calculate it using formulas that we've talked about here. So you have r minus 1 degrees of freedom, because when you have to calculate the standard deviation, you burn a degree of freedom by using the mean estimate. And then you've got the standard area of the mean. This is our standard formula here. And then you scale that by the critical t value using that degree of freedom. And then with that formula, if you happen to be given a 95% confidence interval, and we had you do this exercise, I think on lab 9, you can go from one confidence interval, a 95% confidence interval, to a 90% confidence interval just by scaling by the critical t values. So here's the confidence interval here. This is your 95% confidence interval. You divide out the t value that corresponds to 95. You multiply by the t-value that corresponds to 90, and you get your 90% confidence interval. So you do that, this ratio here tells you how to scale from one to the other. And you know that the less confident you get, the tighter the interval is going to be. And so, we would expect this critical t-value is going to be less than this critical t-value, and it's going to cause that thing to shrink. So that's the idea there. And then if somebody asks you for a particular half width, we've got a formula for that. So you have to you have to ask them what is their preferred half width, epsilon. You have to run a pilot experiment to estimate the variance in the system. Knowing the variance in the system and their desired half width, then you can figure out roughly how many replications we need to run of our sim. There was a question after the class last time about, now we have such fast computers, do we have to really worry about this? And I still today, run for on Army Corps projects where we're placing wetlands to try to better optimize water resource management in like large regions of Texas and things like that. And the simulations that I run take about 10 minutes to run for one replication. So if I have to run 100 replications, that takes a significant amount of time. So it's really important for me to reduce the number of replications. So even with modern day computers, it's still important to reduce the number of reps. Okay, so then we talked about comparing two systems. So I've got a system, an as-is system, and then a proposed optimization. I'm going to take data on those two. When you're thinking about your final projects, it's a lot easier to defend relative performance estimates than absolute. If you say that if you change your system so that you're doing this, then your customer wait time will be exactly 5 minutes plus or minus 30 seconds. That might be fine if you've got a perfectly well-calibrated simulation model. But if it's a sim model that you've only, you spent a couple weeks on, you haven't gathered a lot of data on, you haven't had a chance to do a lot of calibration, then it's hard to take the absolute numbers seriously. So, you would have to, you have to show a lot of other things to show that this simulation model for the as-is system produces all of the absolute numbers you get in the real system. So that's why it's a lot of times it's really hard to say, yeah, my sim says you're going to save, $300,000, and then you actually implement the real thing. You might save money, but you maybe only save $25,000. That's still a savings, but it's nowhere near what you expected from the sim because the sim wasn't well calibrated. But a relative performance estimation, oftentimes you don't need a well-calibrated model because even in a poorly calibrated model, the difference between the as-is configuration and the proposed configuration is still going to be ordered the same way. It's not always the case, but very often it's the case. So even if you're not getting the absolute numbers right, if you just want to know, is it going to be an improvement for my system to go from the system where there's no cross-training over here to the system where we've got cross-training over here, then in that case, that difference, like it might be even on a poorly calibrated system, it may still rank the same way. So relative performance estimates are often more robust to your model not being as well calibrated as it could be. So we got these two systems, we want to compare it. And so we learned about that we can use a confidence interval to summarize those things too. But now it's a confidence interval on the difference between the two systems. So we're taking the standard error of the mean difference, but we still generate a half width. But now we're interested in whether this difference confidence interval includes 0, just like in that regression coefficient example. And we said, based on how you set up your experiment, there's 3 different ways you can use 3 different standard error formulas and 3 different degree of freedom formulas based on how you set up the experiment. If you use a paired design, you use this up here, which basically looks just like the one sample version because you're really just doing a one sample test on differences. There's a 2 sample test. If you can say that these 2 systems have the same variance, then you can pool them together and you get a little bit higher statistical power. So it's a little bit more sensitive. We mentioned there's ways in which you can test whether there's a variance. I think the thing you learn in 380 or 385 is like an F test, but an F test ends up actually being really, really sensitive, and it's really poorly performing if your data aren't perfectly normal. So although we teach an F test when we first start about these things, we never use an F test in practice because it's too dangerous to use an F test because too many things can go wrong. So when you're testing variance, you typically use something like a Levene's test, and then that can say, do these things have sufficiently close variance? And if they do, then maybe you can pool the variance together and you use these formulas. But if you decide that, no, I have to consider these to have different variance, then I use the Welch's test down here, which has this uglier formula for degrees of freedom, but simpler formula for standard error. And that's how I form this confidence interval. So that's what we talked about last time. Now, thinking practically, it turns out that really we never use the pooled variance t-test. This is another one of those things that, well, the first time I teach you this stuff, I'm supposed to teach you both so that I force you to practice, knowing that there are two types of standard error. And so, to appreciate that there's these standard error formulas that you put in and so on and so forth. But it turns out that in order for the conditions to be really well suited to pool your data together, to have this pooled variance estimate, it's rare for those conditions to hold so perfectly. And so you end up running a lot of risk by pooling your variance together, and you don't get that much more power out. So what most people do is they just use the unequal variance test, the Welch's test. You don't lose that much power, and you can make a lot fewer assumptions. It's a little uglier formula here, but that's not that big of a deal. So then that's what you use to form these confidence intervals. So I do want you to know that a pooled variance test exists in case you ever see one, but it should raise red flags because very often you don't have enough of the assumptions to justify using a pooled variance approach. And so everybody uses a Welch's approach. You just assume the variances are different. You do lose a little power, but it's not that much power. Okay, so questions about that? That was some of the newer stuff. Okay, and then, so what do those things look like when people report stuff? Well, people really do report mean differences. So here's a paper where they did a 2-sample test. These were actually paired tests, and so exactly what they're testing doesn't matter too much, but I just want to say how they wrote it out. So, in 2 of the 3 sets, Lilith, which was one particular experimental replicate, they just happen to be named because these are animal models. And they give a t-value, and then NS, non-significant, paired t-test, mean difference, minus 0.071, 95% confidence intervals, and they give these confidence intervals. And this mean difference is just the middle of this confidence interval. And you can see the confidence interval starts negative and goes positive. So the confidence interval includes 0. And that's why it's non-significant. Down here, same thing. Confidence interval includes 0. That's why it's non-significant. But then down here, they've got a confidence interval that for one of these t-tests here, so one-tailed paired t-test, its confidence interval does not include 0. It's purely negative. So in that case, it was significant. P is equal to 0.01. So they're listing everything here, but and that's just to be complete. But very often people just list their confidence intervals. So you got to know the confidence intervals, what it includes and excludes. That's what you're supposed to be paying attention to. And you don't even necessarily need the midpoint. So a lot of times they'll give you that just because people are interested, but this is just the mean value of the confidence interval. So a lot of times they just give you the confidence interval and that's it. So if you have a paired design, then oftentimes the way you summarize it to people is with plots like this. So every one of your groups, you can plot on an x-axis, and then you can plot the 2 treatments for those 2 groups with 2 different label types. So here, it doesn't matter what this experiment is, but they've got these A through J blocks, experimental blocks. And for every block, they ran these 2-wall and 1-wall treatments, and they got a different shift measurement here. So the 2 walls are all the X's, and the X's tend to be on top, and the 1 walls tend to be on the bottom, not everywhere. So here, they were nice enough to show a dashed line when it flips the order, just to show you that there is at least one of these where the order is flipped. And so what you're supposed to take from this is if you were to unpair these, you'd see a lot of variation between the X's and O's. And it would be maybe very difficult to tell whether the X population was different than the O population. So imagine collapsing all these groups onto the X-axis, or onto, the Y-axis here, so there were just X's and O's. Well, you might get a cluster of X's and cluster of O's that are overlapping on the y-axis. And you might say, well, those populations look very similar. But when you expand them out and look at all the groupings, then it's pretty clear that most of the variance is coming from this group factor, from this factor here. This is a randomness being added to the results. And if you control for that randomness, which is what a paired test does, then you see that within every group, it's pretty consistent that the X treatment is larger than the O treatment. So if you look at those differences, then all of the differences are pretty much positive, except for one case there. And so that's why it's nice to summarize paired tests like this. And so if they're unpaired, so they're independent samples, sometimes you just use the box plots like this. Now, sometimes people do paired tests with box plots, but they'll show all their data points like on top of the box and all their data points on top of the box, and they'll connect data points so that you get to see what those groupings are. So, but in this case, that's, this is data from an experiment that did not have pairing. So these are two independent groups, and this group has a lower average than this group. So that's how you summarize that for two sample unpaired. All right, now what we didn't get to last time is what about three or more? So you've got three different configurations or more. How do I deal with that? And so in that case, you have to deal with these ranking and selection methods. And so a ranking and selection method allows you to compare all models to each other and hopefully figure out which model is best. So what we're going to do is for every pairwise combination, model A to model B, model A to model C, model B to model C, each one of those we can ask, I'm going to do a 2-sample confidence interval for each one of those. And I'll say, is there a difference between A and B, A and C, and B and C? And from those, I can maybe then infer whether A is better than both, and so on. But because I'm doing all those comparisons, that Bonferroni test I was talking about comes into play. So you have to count up how many comparisons that you're making. And so in that case, I had 3 systems that I was thinking all pairwise comparisons. Well, that's, 3, it's basically, 3 choose, so 3 systems, pairwise comparisons. So I've got, and it's going to be 3. So that's what we've got the formula here, is that, 3 times 2 divided by 2 gives me my 3 comparisons. So if I had more, if I had 10, and now I want to do every pairwise, well, now I've got whatever, 10 times 9 divided by 2 times 1. So that's a much higher number. So for that number of comparisons, we talked about Bonferroni correction. For each confidence interval, I need to set the level of that confidence interval to the 5% I want for the family by this number of comparisons. So in order to get 5% at the ranking level, looking at everybody, looking at every individual comparison, I have to crank my alpha down to really low level. So I effectively need a really high confidence interval for each individual comparison in order to get a moderate confidence interval at the family level. So this is going to really impact my statistical power, because it's going to be very difficult for me to notice things between individual pairs, but I have to do that correction to prevent from noticing a bunch of false positives accumulating at the family level. So this is sort of not a great way to do ranking and selection, but it is a brute force way that works if you have few comparisons. But if you have many comparisons, this isn't ideal. So what are other ways to do that? So, hopefully you've talked about that in ANOVA in previous classes. So an ANOVA is a one-shot way to do this. So if you've taken all of your data, you don't want to run any more experiments, you just want to analyze your data, then you do an ANOVA with what's called a post hoc test. So you run your ANOVA. So in this case, I had these 5 groups. So you can imagine this is my as-is system. This is my system with cashiers. This is my system with robots. This is my system with kiosks. And this is my system with purely mobile orders. I don't know. You could come up with 5 different systems, and I want to compare them. And the first question you have to ask is, did I make any difference whatsoever across my groups? Are there any differences in my groups? And that's what the ANOVA tells you. So the ANOVA will give you these graphs, and if an ANOVA comes out with a significant p-value, p less than 0.05, that's telling you that one of your groups, at least one, is better than at least one other group, that there is a performance difference among your groups. But an ANOVA doesn't directly tell you which one it is. So you can, that's when you run these things like, this is called a Tukey's HSD post hoc test. Another one that's popular in engineering is the Fisher's LSD post hoc test. It has a little bit, it has an inflated type 1 error, but we're okay with that in engineering, as I've talked about before. But the idea here is it puts these grouping labels. Notice this A, B, B, and B. Those are added by the post hoc test. What the post hoc test tells you is that everything that shares a letter can't be told apart in any statistically meaningful way. If anything it does not share a letter, then those are statistically different. So what this tells me is that I can't tell a difference between these 2 groups that are labeled A, but I can tell a difference between either of these groups and either of these groups that are labeled B. But this middle group has both an A and a B, which means that I can't sort it in any, like I don't know if this should be grouping with B or should be grouping with A. It's probably something intermediate, but it is possible if I took more data that this thing might actually, like I might have gotten lucky and that's why it's up here. And if I took more data or did another run of this experiment, this group would plummet into the ground and it would be like the lowest of the A's. Or if I did another experiment, it could be up here with the B's. I don't know. This thing is within chance to be in either group. So I can't tell the difference. But if you're just looking for, I need I need to know, I need a group where I get is going to be in the highest. Like I need a recommendation that is going to maximize this. Well, you can tell your stakeholders that, well, these 2 groups, I could not tell the difference between them, but I have evidence that they are definitely higher than these 2 groups and maybe higher than this group, but it was only a trend. And so maybe then your stakeholders okay with saying, all right, since you can't tell the difference between these 2, I'm going to implement this one. Or maybe I'm going to do another study where I just focus on these and I exclude these from the study, and that makes the next study cheaper. So that's how we interpret a post hoc test. We look at these letters, and the letters allow us to sort out what differences were found by the ANOVA. So, and we have tools inside Arena that do effectively this for us. Now, the downside of this is that, for one, in order to do all of this ranking, you need a lot of data, you need a lot of power. When you may only care about whoever's at the top or whoever's at the bottom, and you don't care about sorting the rest. The other thing is you don't run an ANOVA in sequences and stages. It's not like you say, I'm going to run 10 replications, I'll do my ANOVA, I'll add another 10, do another ANOVA, add another 10, do another ANOVA. Then you start inflating your error because you end up using the same data in multiple comparisons. So there are additional techniques that I went into in detail in the last lecture at the end that we've kind of skipped. I'm not going to quiz you on those, but if you're interested in those, you can take a look at some of those. But basically, there are other techniques that allow for sequential sampling that allow you to start with a pilot number of replications and then add new replications iteratively until you reach a ranking standard. And then usually those aren't focused on sorting the whole group. They're focused on just finding the top or finding the bottom group. And so and that's kind of what we're summarizing here. And so I talked about the process you could do if you want to do this manually. Fortunately, the process analyzer in Arena does a lot of this stuff for us. So by now, you've done, you've played with the labs, especially if you're in lab 10. Some of you have been working with the process analyzer. It's also one thing you can use. You don't have to use, but in homework J2. And so here you've got a bunch of scenarios, and this says 3 scenarios you like to compare. ran a certain number of replications, 5 reps of each of these scenarios. You have controls that are set. So these are the differences between the scenarios, and you have different costs that come out of these scenarios, and different numbers of rejected calls. And so if you run one of these box and whiskers charts from a process analyzer, it's colored like the post hoc test was labeled. And so you can tell it, but it doesn't sort everything. You basically tell this process analyzer tool, show me the best or show me the worst, show me the highest or the lowest. And it will color the things it can tell the difference between. So it's saying here that blue is significantly different than red, but I can't tell the difference between any of the reds. So if you're looking for the lowest value in your optimization, either one of these is statistically arguable to be a reasonable choice. If you're looking for the highest, then scenario 1 is definitely the one you should pick. So that's how we read those colors. And that's basically just a simplified version of that post hoc test, but it's because they only focus on highest and lowest, it can make the inference with less data. All right, any questions about the interpretation there, how to read these? Okay. And then the other tool that you're going to see in Lab 10 is OptQuest, which takes this to another extreme where you don't even know what scenarios to compare. You just tell it what the degrees of freedom are. What are the different ways you could change the sim? And it will then start with some initial configuration of your system, and then it will run that initial condition and get some performance out. It will then randomly change all of that to another scenario that it comes up with, and it will run enough replications to get a performance metric out of that. It will then randomly change again, and it does this over and over again. The reason why you see a red line and a green line is you can also define constraints. So you can say, you're going to be randomly changing my system. Occasionally, you might get me into a point where you've actually violated a constraint that I must meet. So it might be like, change whatever you can, but you have to keep the maximum waiting time under a particular amount. If you don't do that, I can't even consider scenarios where the max wait time is over that amount. Or it might be a constraint on the inputs. It might be a constraint on the actual controls here. Like, some flexibility in the controls, but the sum of these two numbers can't be more than this. The budget that I can spend can't be more than this. And so what this is saying is that it happened to start on one that violated a constraint, and then it tried to get out of that infeasible region by trying different numbers. And so it jumped to another one, and it got even farther away. And then here, this gap is it sort of tried something else that jumped back into the feasible region. And so these are all reasonable ways to configure the system, and it's trying to minimize. And so it's randomly changing the system and then trying replications, and then eventually settling down on some optimal value here. And then you can drill down into any one of these points you can drill down into and say, what was the system configuration when you got this value? And so it does a lot of work for you. It's a little buggy to run though. So a lot of times you might find you open up OptQuest, save frequently because it might crash. This is a third-party program. You can find OptQuest is not made by Rockwell Automation. It's licensed by Rockwell Automation. So if you use other simulation tools, you'll find that they also use OptQuest. So it's a tool you'll see in other programs as well. Okay, so I think that catches us up. Any questions about the mini systems ideas here? Okay. All right, so last unit, VRTs. Variance reduction techniques for simulation. So we've talked about so far, this standard error of the mean. Now we see how confidence intervals are used, analyzing results. We know the confidence interval is built upon the standard error of the mean. The standard error of the mean's got two components. It's got a numerator to denominator. We've been focusing so far on the denominator. We've been focusing on the number of replications. So if you want to have tighter confidence intervals, you have more replications. But we know the replications cost money. So how do we get tighter confidence intervals without more replications. And so we're going to try to focus now on the numerator. That's the variance that we're reducing in variance reduction techniques. So how can we use clever experimental design to reduce the unexplained variance here so that a variance that this summarizes is meaningful variance as opposed to variance that's just introduced by our experiment? So that's what we're starting with. We're going to talk about these two today and these two in our last, kind of meaningful content lecture next week before Thanksgiving. And all of these are different ways. There's a bunch of different ways to do VRTs. These are the common ones that we typically start with. And we've already talked a lot about the first one, common random numbers. This is just pairing your simulations up together. But in this case, we're not necessarily pairing, a system from the real world with this system. We're going to be pairing simulations together by their random number seeds. And that helps guarantee we run the same customers into the two different, even though the systems are implemented to do different things, they get the same customer inputs because they've got the same random number seeds. And that reduces the variation that's just due to having different customers. So I talked about this last time. I kind of introduced this idea already. If we're looking at the difference between system A and system B, if you just do a little probability, a little math on this variance formula, you get this down here. This is the variance of system 1 divided by number of replications, variance of system 2 divided by reps, and then this is a correlation component. So this is a correlation coefficient. So if the two systems are highly correlated, this number right up here in the numerator, rho 1, 2, that will be large. If they're negatively correlated, it'll be negative. It'll be like -1. And then if there's no correlation, if you can get any value for one for any value of the other, then it'll be near 0. So the idea here is that baseline, the variance in the sum is just the sum, the variance in the difference is just the sum of the two variances scaled down by the number of replications. That's our baseline. And we want to say, can we design an experiment to do better than baseline? And how do we do that? We try to introduce correlation between our systems. So what we say is, if we know that sometimes some customers are going to lead to longer wait times and other sets of customers are going to lead to shorter wait times, then we want to avoid the situation where we've got one system's got a bunch of customers with long wait times just because of the nature of the customers, and another system that just happened to luckily get customers with shorter wait times. So we're trying to put things on a level playing field. And so now we're saying we're going to run the same customers into both. And if we think that there's an intrinsic property of how these customers arrive, they all clump together, they all order the hardest things to make, those sorts of things. If there's some property that makes this a hard group of customers and this an easy group of customers, then we can, if we run the hard group of customers into both systems will show longer wait times. That's the correlation that we're looking for. And if that correlation exists, then this little trick of introducing correlation will reduce the variance in their difference between the two systems. That's what we're looking for here. And that's what I'm summarizing down here. But we have to use that paired difference t-test. So we did this in the first lab, that muffin baking simulation. Each one of you have a different, you have one of 10 different digits ending your ASU ID. Based on that, we gave you a different data set of customers. And then those same customers, you ran into two systems, a system that had one batching policy and a system that had another batching policy. And so this is the batching policy greedy. This is the batching policy nearly full. And then each dot here, this is, if your ASU ID ended in like a 0, you might have gotten this for greedy and you might have gotten this for nearly full. And so every policy was replicated on the same set of customers, and then we used multiple sets of customers, and then we could connect them together. So this is, like I mentioned, this is one way that you summarize paired data, is that you show this population and this population separately, but then you connect the two so you see which ones are in the same block. And this, even though in this view there was overlap, so that suggests some ambiguity. It kind of looks that this one is the longer group, the group that takes more time, but there's overlap. And so maybe that's just due to random chance. But when I pair them, I see that for every customer group or for every batch group that comes in, then these are all positively sloped. So for every batch, it just happens to be that nearly full takes longer than greedy. It's true that there are some greedy that take longer than some nearly full, but the difference there is explained not by the policy, but by the input. So by pairing by inputs, we've subtracted off the variance due to input. So the only variance left over is the variance due to policy. We've controlled for input variance, and that allows us to have a smaller output variance so we can get a tighter estimate of the effect. That's what we've done here. And we could summarize that. I mentioned that sometimes you like to summarize it here. Here are the digits at the end of your ASU ID. And here are the greedy policy is the open circles. The nearly full policy is the stars. And you can see in all of these cases, the stars are above the circles. For some groups, there's very little difference. For other groups, there's a lot of difference. But for every group, you get the stars on top of the circle. And that becomes much clearer when you take these differences. And so you put the differences, you can basically build a confidence interval on differences. 0 is going to be outside of that confidence interval. So we can conclude that one system is definitely better than another system. The textbook does a similar example. So here's an example here where they did independent sampling. So they just happened to pair these up randomly. So they had a queue with one server and a queue with two servers. The one server is dark, the two server is light, and I think they were measuring waiting time, average waiting time here. And so when you don't pair them, then it like when they don't have the same customers coming in, then it doesn't make sense to pair them. It's kind of randomly paired, and you find that sometimes the blacks are above the whites, and sometimes the whites are above the blacks, or the filled are above the solid, or the open. But when you pair them so they get the same customers, then you notice a pattern. And you notice that the pattern that you'd expect, that the waiting time with one server tends to be longer than the waiting time with two servers. So what they're saying here is these are underutilized servers. These servers are not near capacity. So there is a small effect of having another cashier, of having another server. But, and we could not see that effect when we didn't pair them together. In fact, here, there's really large differences just due to random chance because this pairing, the one that's filled, the one server happened to get lucky enough to get really long gaps between inputs. But this one had really short gaps. So they kind of flipped things from what we'd expect. But when we put everything together, apples to apples, then we see that there's always a benefit in terms of waiting time to having two servers. It's just a small benefit. So we could not see, we've made the test more sensitive. We've made the confidence intervals tighter so we can detect smaller differences. An undetectable effect because of the high variance in input is now detectable because we've subtracted off the variance due to input. That's the magic there of adding this correlation. So how do you do this in Arena? So in Arena, you pull in this elements panel. This is one of the ones that is not included by default. So you have to right click and say attach panel. You pull in these elements and it gets these old school blocks from like the earliest version of Arena that are still there to do these sorts of things. And you pull in this seeds block, you drill down into the seeds block, you can add a seed. We talked about this in the beginning of the class or before the midterm. You can name it whatever you want, like inter-arrival time stream. You can leave the seed value blank. It doesn't matter what that is. But then under initialize option, you select common. So that ensures that any sim that runs, and so like when you're in your create block, you'll end up tying this, and I'll show you how to do that in just the next slide. In your create block, when you're saying I've got exponential inter-arrival times, You're going to say those exponential interarrival times, I want you to draw those numbers from this stream, from this, using this seed configuration. And it will use this common here, and that will ensure that if you run another simulation where it's configured the same way, then it will use the same random number seeds every time. So I think with common here, it basically takes whatever the random number seed is from the previous replication and adds 100,000. And so every replication, you're still changing the seed, but you've got exactly the same seed at the beginning of every replication. And you can close down Arena and reopen it with a new model. As long as you've configured this the same way, you'll get the same random numbers going into your create block. So, and that's what I'm summarizing here. You just open that thing up, you set that to common, and then common, like I was saying, sets the next replication to be whatever the seed was for this replication plus 100,000. So it guarantees that you know the seed at the beginning of each one of those replications. So you go into your create block, into this expression. You normally in expo, so you have to force this to use expression. You can't just select exponential. And normally with an expo would have one argument, the mean time, the mean interarrival time. But the second optional argument here is that name of that stream that you created in this seeds block. And that's how you tell for when you create arrivals, I want you to draw the numbers from my common random numbers stream. So that's how we do that in Arena. That's I'm just summarizing there. Questions about that? They're pretty clear, the basic idea and how you would implement that in Arena. So the downsides of this, you have to keep, does it always work? It doesn't always work because it may be that your two systems you're comparing are so different that a hard input for one system is an easy input for another system. And if that's the case, then doing what we just said is actually going to make things worse. So before you use common random numbers on a large number of replications, you should pilot it for a small number of replications. And so you run a little pilot experiment, and then you measure the variance in your system 1, the variance in your system 2, and the variance in the difference between those systems across their pairs. And if the CRN magic is working, then the variance in the difference should be less than the sum of the two variances. If it's not, then that indicates that you're in a situation where your two systems are so different that CRN isn't going to help you, and you should just run them independently. Okay, questions about that? That only works if you manage to get correlation between your systems. Okay. All right. So last thing I want to cover today then, and then we'll do the last two VRTs next time, control variates. So what's a control variate? So I already hinted at this a couple lectures back ago, is that sometimes you know that there'll be sources of variance on your output that are due to things that you can measure in your system. And so here's an example from, not a simulation example. This was a study where they were trying to see what is the effect of publishing with top authors on the first 3 years success of someone in the 20th year of their academic career. So they're trying to say that if you early in your career, did the people you publish with affect how successful you were later in your career? And so their key predictor there was, we're going to look at the literature, and for everybody who published, we're going to see who they published with. So that was what they're interested in, predictor of interest here, top co-authors. But they realized that there's a bunch of other things that affect success in the academic industry, in the publishing industry. And they might be, what institution you're at, general productivity, how many citations your papers get. And they wanted to say, we're interested in the general effect of this predictor of interest after you subtract off the extra variance caused by these things. So even though they weren't interested in these things, which other people already showed were a were a contributor to success. They had to build a model that accounted for all four of these predictors. And so that's what we see here. And so this is for this particular field, cell biology. What you see here, this line 0, this is their reference line, because all of these things that look like frog eyes, those are confidence intervals. And so the dot is the center of the confidence interval, and the little lines on the other side are the edges of the confidence interval. And so this is saying, is there a significant effect of each one of these things? And if it crosses 0, then that would mean that you can't tell if there's an effect. But all four of these frog eyes are to the right of this 0 vertical line, which means all four of these have a significant effect. These are basically regression coefficients. And all four regression coefficients have a positive slope. So they all four have an effect. Just notice, though, that the one that they were interested in, top co-authors, it's got the smallest effect. It's got the flattest slope. So if they hadn't had controlled for these extra sources of variance, then the confidence interval here would be very large, because it would say, well, we're trying to look for a signal due to which authors you published with, but because we haven't accounted for all of these things, then there's all of this extra variance, like there's just extra noise in the system. And so by also measuring these things and how much noise they create, they could create a tiny confidence interval here and measure the small but apparently significant effect of the thing that they were focused on, top co-authors. So this is the motivation for what we're trying to do here, is that if you can measure things that you know will have an effect on your outputs, you should, because then you can subtract their effect from the output off, so that what's left over has less variance, and it'll be easier to zoom in on the thing that you don't know about but care about. So that's what they did here. And here is our kind of simulation motivation. So imagine you have you're trying to, you've got a bunch of solar cells on the top of a large structure on ASU's campus, and you're trying to experiment with different ways you can operate those solar panels. So it turns out that the inverters that are underneath these solar panels, there's a bunch of parameters that you can tune that end up making the solar panels more or less effective. And those parameters, the way you tune them depends on whether clouds are going over. There's all sorts of things. So it's a complex optimization problem. So imagine that you wanted to test two different ways to operate the inverters underneath these solar panels. And so you're going to use a simulation for that. Now you look at a bunch of this data, and it turns out that the solar panels, their efficiency is affected not only by these inverters, but how hot it is outside. And so you might want to run an experiment where, if this is a real world experiment, not a simulation experiment, you might want to run the experiment with your inverters in one configuration on certain days and another configuration on other days. But you might have huge variance. Like it might just be that when the time you got to run the experiment might have been in the springtime for treatment A, but in the summertime for treatment B. So now you're going to get a huge effect of temperature. So the idea here is, well, temperature is generating variance in output that's going to make my results more noisy, but I can measure temperature. So if I know that the output Y is correlated with temperature, then I can subtract each day's output by an amount proportional to how weird that day's temperature was. So if I have the mean temperature, was this a hot day or a cold day? And that allows me to adjust for the variance that has been added due to temperature, so that I can zoom in on what I actually care about, which is how I change how the inverters are running. So how do we do that? And this is what's known as control variates. So the idea here is, this is my output Y of my simulation, and this could be applied to any system. It could be a real system, could be a simulated system. So I've got an output Y, and I've got something that I can measure like temperature X, where I happen to know the average value of that temperature. So I know the average temperature over a year, and I know the temperature of the particular day. So I can calculate this expression here. So I'm just going to calculate this z, and this z is what we're going to end up calling our control variate. Now, this z is just subtracting this amount from this. So I've got this variable c that I haven't set yet, but it's just a number. And so I found to say, well, what if I calculated this? Well, what's the expected value of this? Well, the expected value is z, is just the expected value of y minus, and you can push through the arithmetic here, things that cancel out. So the cool thing about this control variate expression is it has the same mean as the output I want. So the output's got a certain mean and variance. The control variate has a certain mean and variance. This expression changes the variance, but not the mean. So that gives me the idea that if I want to estimate the mean of y, I can just as well estimate the mean of z, but I do that with a different variance. This is where I get the lever to reduce my variance, so I can get a better estimate of y. So I'm using z's data to estimate y because they have the same average. That's the idea here. So now I just need to figure out what's the best value of c that maximizes the effect here. So I'm going to do a little calculus. So if I calculate the variance of my control variate, you work through the probability math, you get this expression here. And this expression, you can just say, well, I want to minimize the variance in z. So I'm going to take the partial derivative of this thing with respect to c and just see what comes out. And I get this expression. And this expression says it's the covariance of x and y. So this is how well they correlate. It's another expression of correlation divided by the variance of x. So if they're uncorrelated, so if x and y have no relationship whatsoever, if I was wrong and temperature doesn't have an effect on the output, then this will be 0, c will be 0, and my control variate will just be equal to y. It just falls out. It degenerates to just being y. But if there is correlation, then this thing's going to be positive and subtract it off. If there's negative correlation, then this also works. So any direction of correlation, positive or negative, this is going to work. It's going to minimize that variance. So let's see how we can do that in. This is basically the idea here. The variance in z is going to be the variance in y minus this amount here. So how does that work in practice? So here's a really simple example rather than running a big full simulation, but you can imagine this is like a simulation. Imagine I have this x input here. So this is like our temperature. It's uniformly distributed between 0 and 1, so its mean is 0.5. Imagine that I have a simulation which I'm just going to substitute for the exponential function. So I'm just taking the exponential of x. But you can imagine this is a simulation. I run a sim when the temperature is this way. I run a sim when the temperature is this way. So then I get an output, and I want to say, what is my mean of this output? So like in this case, what is my mean value of the exponential of x when x is distributed between 0 and 1? So if I want to do that expression using the math here, I'd expect to get exactly this value using this integral, this ugly integral here. So I want to do that with simulation. So I'm going to draw a bunch of random temperatures or random inputs here. I'm going to generate a bunch of random outputs. And then from those outputs, I can calculate a mean and a standard error of the mean. That's here, mean, standard error of the mean. So how do I reduce my variance without adding more replications? I don't want to run more than 1,000 replications. I'm going to generate that control variate. So I'm going to estimate my variance in temperature, my variance in x, and my covariance between x and y. Turns out MATLAB's got a function that does covariance for me. So that's what this expression is doing here. And MATLAB, of course, can calculate variance. And so from there, I get this variance and this covariance. So there, I use the formula that I derived on the previous page to get this number. And that allows me to build a new random variable that is just all these outputs minus this number times this difference. And that's what I did in MATLAB here. Now, if I do the mean of that thing, I get 1.72, which is close to that mean. And if I do the standard error of the mean of that thing, I get 0.002, which is an order of magnitude less than that standard error of the mean. So I didn't do any more sampling. I used the same 1,000 samples, and I've now drastically reduced the variance in my output. That's the control variant. By using knowledge of the covariance between an input and an output, I can then subtract the effect of the confounding variance, the confounding input. I can control for the noise that I don't care about. I can remove that so that whatever's left over is just the stuff I care about. And that's what we did there. Questions on that? Variance reduction. Okay. So then likewise, if your boss tells you that that they need you have a confidence interval that they want to be so big, this approach, if you instead figure out the confidence interval, do you run those pilot studies on the confidence interval on the control variant, what you might find is that you might have needed 1,000 replications to get the stakeholders' desired confidence interval. But now with the control variant, you only need 100 replications to get that confidence interval. So that's another way you can use this. Okay. All right, so we just covered CRNs, which we've been kind of reviewing all semester iteratively. We talked about control variates. That's this new approach, which is very closely related to what's going on behind the scenes in multivariate linear regression, which is a more advanced statistical topic, but it's doing basically the same thing there. And then next time we're going to talk about antithetic variates and importance sampling, and that will wrap up all the new content that we have in the class. So let's close here. Let's get you out of here a few minutes early. And the short answer here, it's another one of those things where I'm just going to be, it's a short answer, but I'm going to expect just one of two possible answers. And that filters out people that are just sitting in dorms waiting for this thing to pop up right now. And so for common random numbers to work, do you want positive correlation or negative correlation between your two systems? So for CRNs to work, do you want positive or negative correlation? So on one of them, CRNs will be a major reduction in variance. On the other one, it'll go the wrong way. So those are the two answers I'm looking for, either positive or negative. And that's it. So otherwise, we'll see you Tuesday for the last lecture of the class, really.